So for those of you which have not heard about it, maybe a brief introduction, but then I also will try to focus in on the brain, because you're all brain experts, and we have been working very closely with the Allen brain team and with efforts that we are connected to via the CFAR multi-scale human efforts that's funded out of Canada. And we also had initial contacts with the Yiddish brain, and we would love to continue this collaboration. But we have, compared to some other organs, we have very little data for the brain, and I think we can change this together. And so again, for those of you which have not heard about the Human Reference Atlas, it's a effort that now unites researchers from around the globe, from many different projects funded by the National Institutes of Health, by European projects, by the Sean Zuckerberg Foundation, by Canadian institutions, and now also the Charité Stiftung. And many of these specific atlases, these consortia, for instance, the Allen Brain Atlas, they would focus, for instance, on the brain. And in the interactive visualization, you could click on that link here, on the brain link, and you would get to see all the different efforts that are actually focusing as a consortium on the brain. And there are now more. This is from a paper that's now 3 years old. And back then, those were the efforts that had engaged with us, had started to discuss how to best bring brain data into this atlas that is a multi-organ atlas. So while you all have a skull to use for registration, potentially, you won't have that for the heart or for the intestine or for many of the other organs. And the organs are quite different, and they're very different function, as you know, but interestingly, no organ can do it alone. So you really need the collection of all the organs to have the oxygen, to have the nutrients, to have the brain power to make us work. And even though I have $0 for brain work, I am very interested in mapping and ultimately also mapping the brain. This line of research, which I'm going to present here, comes from a long line of mapping science by itself. Some of you might have encountered the Mapping Science exhibit, which maps science, technology, education, job market data at a large scale and shows how that evolves over time. You can then use these maps to overlay your own career trajectory. You can try to understand developments in your own field of research or identify funding opportunities if this is what you want. Ultimately, all of these maps have to be multi-scale, just like the map of the human body, which is shown here as somebody in a VR environment encountering the female Human Reference Atlas 3D object, and then zooming into a kidney, down into the renal corpuscle, into podocytes, into proteins, ultimately. So on the Human Reference Atlas side, which we have been working on for now 7 years, we have quite a few papers that you might have encountered. And I'm glad to say that all of this data is open source, open code. The recent Nature Methods paper is a resource paper with 50 different software artifacts that all work together. So that by itself was a huge community effort. And then, of course, you can apply science of science studies to study differences in astronomy, in biology, here the biomedical domain, and physics. So these are big projects, big science projects. And it's very interesting how they unfold in an astronomy setting, how they unfold in a physics setting, where you have big infrastructures such as CERN, which are now also used to map entire human bodies and entire human brains and kidneys, etc. But then in the biomedical domain, it's interesting how you can use big science to make entire consortia work. And it's non-trivial to get them to work. It's a socio-technical challenge, and visualizations can help. So we are looking at this from the visualization angle. In 2015, we published brain self-portraits, as we called it. And the idea was to find out how far can data visualization help to really help our brains to understand itself. Not easy. It's a highly complex organ. And so, again, even though I'm not receiving funding for any brain data connection or mapping, I'm very, very interested in that one organ. And to be honest, it would have been really sad to have a human reference atlas without any brain data. I think that would have been wrong. So I'm glad that we have the Allen Brain team and other teams that have started to work on this challenge to register data into the brain. And the previous speaker also very kindly and nicely explains that this is really important to understand where certain types of data come from spatially, but then also how they can be understood in a multi-scale manner from the tissue down to the cell level to the protein biomarker genetic level. So again, for those of you which have not heard about the atlas, this is our definition of an atlas. It has to have a reference system, just like in the US, the zip codes actually give you an XY on the map. And then you have to have a way to map new data into this reference system, not just for brain, but for all the organs that are the vital organs currently. And it has to be digitally available. So while most atlases don't have an API, they are books typically, here you have an API and a knowledge graph that connects very easily to other knowledge graphs and helps create an ecosystem of free open data, code, and tutorials. Again, in the last 7 years, many of us have worked hard to agree on terminology. So here in box 1 on the right, you have key terminology. And again, the most recent paper is a community effort with more than 270 authors documenting what data and code now exists for this Human Reference Atlas. And you're very welcome to try it out. In our world, we have so-called ASCT and B tables, so anatomical structure, functional tissue unit, cell type, biomarker labels, to capture this multidimensional scale of the human body. And so here you see one exemplary screenshot of a subset of the kidney table. So from kidney as an organ, which is in the urinary system, which is in the body system, you can go down into the nephron and then into smaller and smaller anatomical structure. You have cell types that are located in these anatomical structures, and you have biomarkers that have a typology tree that are used to characterize these cell types. In the HRA, we have proteins, genes, lipids, and metabolites, proteoforms as biomarker types. And then if we go over to brain, which is now in version 1.7, and some of you might have contributed or reviewed this table, you can then go to this link down here in the interactive visualization and start exploring that data, or you can download it and check out all the anatomical structures that are covered, which is quite a few, 188 anatomical structures, and then 622 total cell types, also thanks to Richard Scheuermann contributing to this effort. And then in total, we have 622 cell types and 828 total biomarkers. So it would be very interesting to compare that with some of the data that have been presented here and see if there are others that should be added. Again, this is an iterative process. Version 1.0 had much fewer ASCTs and Bs. And then over time, these get validated against literature, against experimental data, and of course, against expert knowledge. Life unfolds in 3D. So we have reference organs in 3D modeled using the Visible Human project data that the National Library of Medicine has made available for male and female, not just male. You also see that there are some sex-specific organs. And you see on the right-hand side the two brains. One has been fitted for female and one for male. These brains come from the Allen Brain Reference Atlas from Ding et al. In 2016. So we did fit those in, and then we are registering new tissue into these organs. Recently, we also started a very exciting collaboration with a group of dentists, and they are bringing in a lot of mouse anatomical structures. And this way, we also have mouth microbiome and oral cavities, functional tissue units. So if you now have a 3D reference organ, you can start registering tissue blocks into them. Here, these white blocks, they're basically cuts that were cut from organs that couldn't be used any longer for transplantation, or they are tissue blocks that came from biopsies, but these are too large, which you see here, they're typically just cut from entire organs. So here you have the heart, kidney, and lung, and the tissue blocks registered into the male version. So as you see, in some areas we have two. So we have a left and a right kidney, and we have that for male and female. So 4 kidneys in total, and it matters. So we need to understand where this tissue data came from, if it comes from the upper lobe, the lower lobe. We want to understand if there is a gradient as you go downwards, the kidney, etc. And so right now we have data registered from 16 consortia, 578 donors. We have a lot of extraction sites and many, many tissue data sets linked to these registered tissue blocks. And so again, if you have data and you would like to try this out, the registration user interface is fully available for free. You can then share the resulting JSON file with us, and we can make it available in this user interface so that others can find your data and can use your data more easily. For the brain, we recently collaborated with the Allen brain team. We re-registered 209 tissue blocks from a recent paper, and you see them all here in the left hemisphere of the male brain. They will be flipped and mirrored as per protocol. This is the next step, and then these blocks currently just link to the paper. But they will soon also link to the respective raw data sets on the data portal where they are hosted. So we are not keeping the raw data on the humanreferenceatlas.io website, but instead we are pointing out to the many different data portals that hold this high quality data. If you have data registered, then you can start computing which cell type populations exist for which anatomical structures. So if you have a tissue block which sits on 2 anatomical structures, maybe 20 here and 80% here, you can start based on the anatomical structures and the cell types which are typically found in those based on the AST+B tables. You can understand what cells and how many of them are in which anatomical structure and what biomarker expression values they have. So here for a number of male and female anatomical structures, not just at the organ level, but at the anatomical structure level, you now have cell type populations. And this is at the most abstract or most general cell type label level. But you can, of course, go into much more granularity if you have such granularity in your cell type annotations. Again, all of this data can be accessed online, and there's also now a new bioRxiv preprint on this. In our work, we are focusing also into function. Many of the speakers mentioned that function, in addition to structure, is really important to understand. And so here you have functional tissue units that exist across more than 10 organs. In total, we now have 27 of these functional tissue units. And we are zooming in on those to understand how cell type populations and biomarker expression patterns change as we age, and also as disease comes in. And so again, if you now go to the IFTU Explorer, you can pick one of those 27. You can see a 2D illustration of it. You can hover over any cell type. You get to see the cell type and the biomarker expression value matrix here. And if you have multiple data sets, for instance, for younger and older populations or healthy and diseased, you can very quickly see the difference across those. Some of you might have spatial data. So if you have an X and a Y and maybe even 3D data, a Z, and a cell type or a cluster label assignment, then you can go to the Cell Distance Explorer, CDE, and start to render distance distributions from certain cell types to endothelial cells. And endothelial cells are interesting on our end because we are trying to use or to see if we can use the vasculature as a common coordinate framework for the healthy human body. And so here you see the distance distributions for different cell types in this particular intestine data set. And it looks actually also quite beautiful if you zoom in. And so again, if you have your data in this kind of format, you can upload it and just generate these distance distributions. Some have also now used this Cell Distance Explorer for the subcellular data they have, and that is also possible. Here you have different published data sets that we run through the Cell Distance Explorer. Some of you might be particularly interested in tumors, here in colorectal cancer. Some of you might be interested in other types of tissues. And again, you can do this for your own data. When you have a connection between vasculature linkages and FTUs, you can start to create these kind of butterfly maps, we call them, because you can put yourself in the middle and take a selfie. But here you have the male and the female. You have the functional tissue units, which are also shown here as illustrations, and you have the vasculature connecting these FTUs from the heart, which is here rendered in 3D, to the FTU, and then all the way back again, sometimes going through multiple organs. And you can then see the multiscale nature of that process. This is the beginning of the vasculature common coordinate framework, which we believe will work potentially the best for having a latitude-longitude system for the healthy human body, male and female. There's an entire movie on this if you're interested to know more about it. I don't think I have time here to show it, but it's available online. If you would like to know more about the Human Reference Atlas and how it's constructed and how it's ultimately used to, for instance, predict cell type annotation for tissue you just cut out of a brain or just cut out of a kidney or another organ, I recommend this paper, which just got published. And here you also see some of the main functionalities. So you have a kidney, you cut a piece of tissue, and you can now start predicting what cell types and biomarker expression values will likely exist there for a certain donor population. Or you have a cell type by biomarker matrix, but you didn't register exactly where that tissue block came from. So you can, of course, based on cell types and biomarkers, identify the organ, but what you really want is to identify the location inside of the organ. And that is now also possible for areas where we have sufficient data. So brain is not one of them right now, but we would love to have it in there. Secondary, you can also take a healthy lung and a diseased lung, and you can look at these cell distance distributions, and they are very characteristic for not just disease itself, but also disease states. So we are working with pathologists for different organs to basically have a Boolean logic to characterize that, and then to also use it on H&E data and other data that is more commonly captured in the clinical praxis. And then down here, you have the multiscale nature of functional tissue units. This is work by John Hickey, and he recently also published that paper, and this is for the intestine. And I should mention that the collaboration with Gloria Priobo's team is on lung. So all this can't be done without having experimentalists actually provide highest quality data. Just to bring it back to Berlin, we are very glad that one of our projects got funded now. So together with Peter Robinson, we are interconnecting the Human Reference Atlas with the Human Phenotype Ontology. So some of the work that was just presented on the importance of genetic traits to disease is super relevant, and I hope to connect more. In fact, I will be back in Berlin to collaborate and work more with Peter Robinson's team on November 11 and 12. So if you would like to connect, just send me an email. Also, given your interest in human physiology and function of the brain, you might be interested to learn that there is a new effort that currently has one team only funded, but there will be many more experimentalists and also computationalist teams funded over the next 10 years, I think. So this is a major effort funded by the National Institutes of Health to get us a reference physiome of the healthy human body. And I would be happy to answer questions there as well. So again, please do reach out, connect. I would be happy to answer questions. Thank you all.